Hello, good morning. Myself, I am BSR Botany lecturer. Today, our topic respiration. So, respiration two types: aerobic respiration and anaerobic respiration. Here, when the substance undergo complete oxidation, called aerobic respiration. When it undergo incomplete oxidation, that is called anaerobic respiration. So, aerobic respiration steps you take: glycolysis, Krebs cycle, electron transport. Whereas here in anaerobic respiration, incomplete oxidation takes place. But both in aerobic respiration and in anaerobic respiration, the common process is glycolysis. So the glycolysis means the glucose converted into two molecules of pyruvic acid. In each glucose, six carbons are present, but in the pyruvic acid, each pyruvic acid, three carbons are present. Now the process. Here the first substance is glucose. Glucose converted into glucose 6-phosphate because of phosphorylation. Here the phosphate is added. So phosphate donor is ATP. ATP converted into ADP. For that the enzyme is hexokinase. Hexokinase activated magnesium ion. So magnesium ion is required. So anyway, then glucose 6-phosphate is formed. Glucose 6-phosphate isomerase to fructose 6-phosphate. Glucose 6-phosphate, fructose 6-phosphate. That process is called isomerization. For that, the enzyme is phosphohexoisomerase. The enzyme phosphohexoisomerase. Then, when you go for fructose 6-phosphate, again undergoing phosphorylation. So, for phosphorylation, the phosphate donor ATP. ATP converted into ADP. So, two times phosphorylation over. So there the process is phosphorylation. So this is the first phosphorylation. Here this is the second phosphorylation. Phosphate donor is ATP. Enzyme is phosphofructokinase. So fructose 1,6 bisphosphate formed. In each fructose 6 bisphosphate, 6 carbons are present. In each fructose 1,6 bisphosphate, 6 carbons are present. Now it undergo cleavage. When fructose 1,6 bisphosphate undergoes cleavage, two products are forming, DAP and GAP. DAP means dihydroxyacetone phosphate. GAP means glyceraldehyde 3-phosphate. Now this DAP, dihydroxyacetone phosphate, isomerizes to glyceraldehyde 3-phosphate in presence of triose phosphate isomerase enzyme. That process is called isomerization. Then this glyceraldehyde 3-phosphate, two molecules of glyceraldehyde 3-phosphate now undergo oxidative phosphorylation. Here oxidation, there is no direct involvement of oxygen. There is no direct involvement of atmospheric oxygen. Here this oxidation is removal of hydrogen. So that is why NAD converted into NADH2. Like that, for 1G3P, 1 NADH2, for 2G3P, 2 NADH2s can be synthesized. So oxidative, not only oxidation and also phosphorylation. Generally for phosphorylation, here ATPs are required, but here the phosphate donor is H2PO4. H2PO4 present in the cytoplasm. Okay, clear? So that is why glyceraldehyde 3-phosphate undergoes oxidative phosphorylation to form 1,3-bisphosphoglyceric acid, that is called 1,3-BiPGA. So as a result of oxidative phosphorylation, 2 NAD converted into 2 NADH2. For that the enzyme is glyceraldehyde 3-phosphate dehydrogenase. Later on, when you go to 1,3-BiPGA, undergo dephosphorylation. This is the first dephosphorylation. Removal of phosphate. When the phosphate is removed, ADP converted into ATP. It means here ATP synthesis takes place. For that, the enzyme is phosphoglycerokinase. The enzyme phosphoglycerokinase. So at substrate level, through reactions, ATP synthesizing, mitochondria is not involving. Hence, that phosphorylation is called substrate level phosphorylation. So this is the first substrate level phosphorylation. Now 3-PGA converted into 2-PGA, just isomerization. For that, the enzyme is phosphoglyceromutase. Phosphoglyceromutase enzyme is required for the formation of 2-PGA. Then again 2-PGA converted into PEP, phosphoenol pyruvic acid, so that there due to dehydration, removal of water. For that the enzyme is enolase. Anyway, finally, phosphoenol pyruvic acid undergo again dephosphorylation, removal of phosphates. So that is why ADP converted into ATP. So their second dephosphorylation takes place. Like that, two ATPs are synthesizing. So for that, the enzyme is pyruvic kinase. Finally, that phosphoenol pyruvic acid undergo dephosphorylation to form pyruvic acid. So this is glycolysis chart. You just observe in the glycolysis chart how many oxidations are there? Only one oxidation. How many dephosphorylations are there? Two dephosphorylations. As a result of one oxidation, how many NADH2s are synthesizing? Two NADH2s are synthesizing. As a result of two dephosphorylation, how many ATPs are synthesizing? Four ATPs are synthesizing. So four ATPs are synthesizing. How many ATPs are consumed? Two ATPs. Here one, two ATP. So minus two ATP. Finally, net when you take 4 minus 2, 2 ATP result, 2 ATP and 2 NADH2. Even this 2 NADH2 also converted into ATP in mitochondria. That is the next process. So this is the glycolysis process. Thank you to all.